Bueno, voy a hacer la presentación en inglés. Well, we have today here Ivan Shpurov. Shpurov.
Yes.
Is that right? Good.
Close enough.
How is it, Ivan Shpurov?
Yes.
Yeah, good. Okay, yeah, so he's going to be talking today about about using physical concepts to investigate collective behavior in humans and in animals. So I'm going to read his short bio. He's now concluding his PhD studies in the Okinawa Institute of Science and Technology. Earlier, he graduated from Moscow State University with a degree in neurobiology. He works in modeling human and animal behavior and analyzing biological data using methods developed in statistical physics and machine learning. His main subject expertise is complex systems, which is a relatively new field of study that can be best described as the application of established physical and mathematical methods to new subject matters. Insect societies can be modeled as active matter particles and cities as growing factual structures. He has published articles on insect and human behavior, as well as some conference papers on neural networks. He speaks English, Russian, and he's now learning Spanish. Well, let's welcome Ivan. So go ahead. Do you have everything set?
Yeah.
Do you have a clicker?
Yeah, I have some sort of speaker attached to me. Can we see the screen now? Okay. Good day, everyone. My name is Ivan Shpurov, and I now work in Japan, but I initially, as it was kindly read to us, completed my studies in neurobiology in Moscow State University. And I guess many of you, since I'm presenting at CINCCO, which is a Center for Research in Cognitive Studies, are studying human brains or cognition, right? Okay, I can see people nodding, so probably I'm on the right track. So when I was studying neurobiology, I learned that brain consists of many parts, and these many parts have a lot of very complex connections of neurons. And when I was learning this, I learned a lot of Latin names. I learned a lot of complex terminology, but still it looked a little bit confusing to me. It seemed like I learned functions of many different parts, but I was unable to understand how this complex system can generate consciousness, how it can generate thoughts, and how can it generate experiences which we all have every day. So in trying to understand this, I started to switch from biology to physics. And as you will see during my presentation, my main topic of study are social insects like bees and ants. And we will see how they can be similar to human brains. And we'll also see how using different physical tools, the tools which have been developed to study transitions in gases or in metals, can help us to have some clarity in this bewildering complexity, which is human brain. So obviously, when I thought that, well, brain is complex and we need to find some new tools to study it, I was not the first person to think so. Physicists have, since the end of the last century, when we have been able to start using computers to record and analyze biological data, physicists started to apply physical concepts to the new stream of biological data. And this line of research was very fruitful. So I would like to just mention a few things, which probably many of you have already heard. So one of them is chaos theory, the great sensitivity of some systems to initial conditions. And it has been found that biological systems are especially chaotic. For example, if you record the human heart rhythm, and if you record the intervals between different heartbeats, then in a healthy human, this is going to be a chaotic time series. And what you can see here is an example of something called a Lyapunov attractor. It's a very famous mathematical system of equations which gave the name to the butterfly effect. I'm not sure if you have seen the movie, but it's actually quite good. And it's the only movie, to my knowledge, which has been named after a scientific effect.
What's the name again?
Butterfly effect. So the chaos theory presumes that you can have a fully deterministic systems, which can be described by a set of equations, But if you change the initial condition just a little bit, the outcome, the trajectory of the system would be completely different. And there are ways to discover such chaotic patterns in natural data. And it has been, for example, shown that when a human heart is healthy, then it follows this chaotic rhythm. But then the human heart is exhibiting some sort of condition like arrhythmia or a similar medical condition, then it becomes considerably more organized. And this chaotic effect is only present in the healthy systems. So another very famous discovery, which also came from the physical sciences. One moment, I think there is a small problem with the, let's hope that the screen is not going to blink, but it still will blink. Anyway, so another famous example is something called a network theory. So if you study, for example, neurons in the brain, and if you record the number of synaptic connections each neuron has to other neurons, and then you plot the resulting distribution, you will observe that it's not a normal distribution, which you would expect for many biological data types, but it would be what is called a scale-free distribution, where you have a few neurons who have a lot of connections, and then majority of neurons who have a very few links. Similar pattern can be observed if we record, say, a number of acquaintances each person has, or a number of links different websites would have. Guess which website would have the most links. Like the most arrows leading to one website from another website. Well, it will be websites like Google and Facebook, right? The ones which are gigantic aggregators of information in the internet. And therefore, if we plot this internet, if we plot the entire internet as a gigantic network, we will observe these hubs, which are very prominent sites, and we will observe a lot of sites which have very few visitors, very few links, but by being integrated in this scale-free network, which has these gigantic hubs, they can still be accessed in an efficient way. So these are just some of the discoveries which has been made by applying these physical and mathematical methods to biological and social sciences. But today we are going to speak about something specific, and we are going to speak about phase transitions and the application of phase transitions to describing human brain and also insect societies. So do we all know what is a phase transition? So a very common one, which you would experience every day, is when you put your kettle and you boil it. The phase transition happens when water becomes vapor. And in fact, in the left side of this diagram, it's the right side for you. In the right side of this diagram, we see a phase diagram of water. So on X and Y axis, we have respectively temperature and pressure. And we can see that if we increase one of these control parameters, either pressure or temperature, the state of water is going to change. And if we look to the other side, we see an image which I generated using DALL-E, one of these new large language models, which shows us crowd, which can be seen as existing in two different phases. In one phase, we have an ordered movement of people. In our phase, we have panic. And this can be seen as two distinct states of matter, just as we can see two distinct states of matter in gas and in water. So how do we define phase transitions, and how do they happen? So the first two terms which we will need to understand in order to talk about phase transitions are order and control parameters. So if we consider the boiling of water, there are two order parameters which we need to take care of. The first parameter is obviously temperature. When we increase temperature, the molecules of water get more energy, they start to move more. Therefore, the aggregate state of the entire system changes. Now, the second parameter which we need to take care about is obviously the pressure, because water boils at different pressures. The boiling point of water is different at different pressures. That's why it's impossible to have a nice coffee high in the mountains. So let's say that a control parameter is something we can tweak to change the state of the system. It's a simplified way to say it, but it's good enough for now. Now, the order parameter is something we measure. It's something we measure to understand like a single number, which gives us an understanding of what happening in the system right now. So a good system to study is a ferromagnetic material. So a ferromagnetic material, which let's say just a simple metal neodymium magnet, the one you can stick on the fridge. Now, if instead of sticking it on the fridge, you put it in a very, very hot oven, and if you rise your temperature up to, say, I think it's around 770 degrees Celsius, the magnet would lose its magnetic properties. So why this happens? So the magnetic dipoles, they are, well, let's for simplicity say that they can point in one direction, one of two directions, either up or down. Now, depending on the temperature, you either have all your magnetic dipoles pointing in one direction, all up or all down. This would happen at low temperatures. Or if you have a high temperature and the system is given a lot of energy, then these dipoles in the system, they're not aligned. Some of them are pointing up, some of them are pointing down. So the net magnetization of the system would be close to zero, and it would not exhibit magnetic properties. So this diagram shows exactly this process. So the magnetization, which we'll measure just as some parameter from 0 to 1, 1 being the maximum magnetization for the material, versus temperature. So once we increase the temperature, the magnetization curve goes down. And at a temperature called the Curie temperature, because it was discovered by Pierre Curie, the material would lose its magnetic properties. And this process of gradual change of magnetization when the temperature is changed is a process of phase transition. And in this case, our control parameter is obviously the temperature, and our order parameter, the one we are measuring to understand the state of the system, is the magnetization. Obviously, real magnets might be a bit complex to study. So happily for us, we have a very simple system, which is called the Drosophila of statistical physics. In the same way the Drosophila flies have been used in biology, this system has been used extensively in statistical physics in order to understand the behavioral phase transition. And this model is called an Ising model, after a German scientist who actually was not particularly successful to study it. It's an ironic story. So this model goes as follows. You have a grid, just a two-dimensional grid, and each side can have one of two values, either +1 or -1. And they represent magnetic dipoles. And if it's +1, it's pointing up. If it's -1, it's pointing down. Look at these arrows. The blue arrows represent, obviously, the -1s. The red arrows represent the +1s. Now, the rules for this system are that different magnetic dipoles, which are adjacent to each other, they want to be pointing in the same direction because it's the lowest energy state for them. That's the state they want to get in. But they have another parameter in the system called temperature. And if the temperature increases, this proclivity of adjacent states to be aligned can be circumvented. So at low temperatures, all four adjacent, if we take any four adjacent sides, at low temperature, they would all have the same value, either +1 or -1. If we increase the temperature, now they're allowed to have different values, +1s and -1s. And obviously, if you sum up their magnetizations, it's going to be some number close to 0, because if you are summing a lot of random +1s and -1s, you are obviously getting 0. So this pictures, the white sides are the +1s and the black sides are -1s. So you see that when the temperature is low, you have big islands of sides which are all pointing in the same direction. And well, you can see a pretty ordered, neat structure. When the temperature is high, well, you can see exactly what you could have seen on an old TV which is not working properly. You see a mess, in other words. All magnetic dipoles are essentially random. There is no structure. Now, if you look at the critical temperature, you can see something slightly unusual, maybe, because you can see a lot of complexity of this picture. You can see a lot of clusters of different sizes, made up of sides with different magnetic moments. And in this critical temperature, a lot of things are happening which are very interesting. So one of them is something called the correlation. So one of these interesting things is a long-range correlations. So imagine that you are putting your Ising model, which is your hypothetical magnet, in a heat bath. So you are setting some temperature, some fixed temperature, and you are allowing the sites to evolve, to change their values. So if you measure the correlations between different time series of magnetic moment changes at different parts of your Ising lattice, and then if you take the mean and if you take all the appropriate metrics to measure the correlation, you will observe that at low and critical values, the correlations between different parts of the lattice are low, because at high temperature, what happens in one side doesn't really affect what happens in another side, and at low temperature, well, not much happens, everything is kind of stuck. But at critical temperature, a small change in this part of the lattice can affect what happens in this part of the lattice. And this means that the correlation are long-ranged, and they can be described with the power law function, as you can see on the right image, which shows the correlation and the decay of correlations of distance, specifically at the critical temperature. So to reiterate, imagine that you are having these 3 Ising models, and then you decide to just change one side, and see what happens. So if you change one side at a low temperature, well, the rules dictate us that this side will probably be the same as its neighboring sides. So if you change it in a way that it is opposing the rules, it will probably change back. Now, if you make this change at the high temperature, well, the temperature is high, so there is very little connection between different sides. So it will probably live its own life and not affect the rest of the system. But if in the critical system you make a small change, then it's possible that this change will affect elements of the lattice which are very far from the original side, which has been changed. And if you think about this a little bit, this high level of correlation, this interconnectedness, already starts to remind us of the human brain, since the human brains are obviously intricately correlated systems, and activity in one part of the neural network can have downstream effects very far away, both in space and in time. This had led scientists to propose what is called a critical brain theory, implying that a vertebrate brain, human brain or any other animal brain, is positioned in the verge between ordered and disordered activity, so-called age of chaos, and therefore is in the state of critical transition. So if you want to learn more about this theory, I would really suggest you to read this book by Per Bak, who was a famous Danish physicist. It's called How Nature Works. It's pretty old and it's not very long, but it has lots of information on the topic. So for now, we'll try to see what is some of the evidence for this criticality in the brain, like what makes us think that the brain is going through a phase transition. So let's look at this picture. What you can see is a correlation network. So imagine that you take this Ising model that we described before, and then you compute all the changes for each side for a very long period of time. If you have this data, you can compute correlation coefficients between different sides, and then create a network of correlation, a correlation network, which will show you that some of the sides are having lots of effect on the rest of the lattice, and some of the sides are not having much influence at all. They're not really correlated with the rest of the system. And conversely, and exactly the same thing, you can do for a human brain if you use fMRI data. So if you work with fMRI data, you know that the basic unit is voxel, which is like a 3-dimensional pixel. And if you take a normal resting state fMRI, and then you compute correlations between all the voxels in the brain, and then do the same analysis, create the same correlational network as you do for the Ising model, then the structure of the correlations, which is this heavy tail structure in which you have some sides with lots of connection and most sides don't have much connections at all, then this structure is exactly mimicking the structure of Ising model, but only when it is at the critical temperature. When the temperature is too low or too high, the system is not imitating the brain at all. One thing. So another way to look at this is to consider something called the connected correlation function. Before we talked about a normal Pearson correlation. Say if you have 2 time series of numbers and then you compute their correlation coefficient, I think that might be familiar to many of you have done any sort of mathematical analysis of data. But another kind of correlation function is called the connected correlation function. In order to compute it, you take the whole system, then you take the mean value of the signal in the system. So if you are, for example, talking about a human brain and you are talking about, say, fMRI data, you have the Bolt signal, which are the fluctuations of oxygen flow to the different parts of the brain. So you take a signal for the part of the brain, then you subtract from all the sides what is the mean signal in this area, and then after this, you compute the correlation between these d-mean series. So to give you an example, you are all presumably listening to me, maybe not, but if we will measure your brain activity or your physiological responses, there will be a great degree of similarity because you are all now synchronized to an external source by talking. But you are probably not synchronized to each other because you are paying attention to me and not to each other. But if we take this mean signal, which is present in everyone, and then subtract it, then we can compute correlations between different parts of this audience as if I was not here. So that is exactly what connected correlation function is. And due to mathematical properties of this connected correlation function, which is shown here for different parts of the brain, and you can easily see that it is an exponential function. And this exponential function is going to cross the zero line at some point. And the zero, the point at which it crosses zero, is very important and is called the correlation length. And what it measures the average distance between two parts of the system, which are like the maximum correlation distance between two parts of the system. The elements in the system which are closer than this distance, they are correlated, they are influencing each other, and the elements of the system which are further apart than this correlation length, they are independent. So at the criticality, this correlation length is pretty high, and it is diverging. What does it mean? It means that if we split a system into many smaller systems, and then we measure the correlation length for each of them separately, then we will see that the smaller subsystems are as correlated as the larger subsystems. So that is a little bit about the criticality in the brain. And it is a very wide topic. And later, if you have some questions, I can answer them. But for now, we'll just move on. And we'll look at something entirely different. And that is something I think many of us faced this morning to get here. And it is traffic in the city. And traffic in the city is a strangely complex phenomenon. And it is also undergoing a critical phase transition. And the control parameter, as you might guess, is the density of cars. So when the density is low, the flow is unperturbed, the movement is fast, and well, everything is good. But when there are too many cars, when the density is high, obviously the total speed of the system is much lower. That is all making sense. But there is going to be a critical state, a critical moment, a critical density of the cars, where the whole system would act as a whole. So let's now see how this happens using a simple model. So this is a pretty old paper from '91. So you can see these are old black and white computer images. But so this system is a cellular automata. The cellular automata are very efficient tools to make simplistic models of different systems. If you heard of something called Conway's Game of Life, then it's probably the most famous cellular automata, which imitates evolution in a computer simulation. But basically, you have a grid, and agents, which are just cells in this two-dimensional grid, they follow certain rules. In this diagram, these agents represent cars or pedestrians who are moving at a maximal speed of 5 if there is nothing ahead of them. But if there is another agent, an obstacle ahead of them, then they stop, and they will always try to move with their maximal speed, but they are also trying to avoid collisions. And when a certain density is reached, obviously the jumps start to form, which you can see are represented on this diagram with the lots of agents which have zero speed. Now, if you look at the flow of agents per unit of road, so May I? So if you have a road like this, and if you measure the number of cars which are passing through the square and keep count, 1, 2, 3, 4, 5, and plot it on y-axis, and then if you measure the density of the square, like what is the average occupancy, what is the average number of cars per hour in the square, regardless of if they're moving or staying, and then if you plot these two quantities, you will observe something peculiar, because initially you see that the flow, the number of cars passing, is increasing as density increases, which makes complete sense. The more agents you have, the more of them are there, the more of them are flowing through. But at a certain point, this trend will reverse, and larger density would mean that the flow is smaller. This means that the density is high because there are some cars which are stationary on the sides. They're just not moving. They're still contributing to the density value, but they're not contributing to the flow value because they're not actually passing through. And this curve is called the fundamental traffic diagram. You can see on the top it's being derived from the simulation, and in the bottom from the real traffic, I believe somewhere in Germany. And in this critical area, you have a maximum variability for the speed of the cars in the system, and maximum variability for the waiting time which it will take you to get from one part of the road to another, and also the maximum flow, so the maximum productivity of the system. So the people who design the actual motorways, they would seek to always be within this critical point in order to maximize the efficiency. But at the same time, this maximization of the efficiency also maximizes the variability, which is not very good for the customers, because you don't want to have a high variability of your speed or your waiting time. You want to be able to plan things. But it's aside. What is important for us to take from this simple jamming model, which is just a one-dimensional cellular automata, that it arises from a very simple interactions. The agents are moving along the road. They are trying to avoid collisions. And when their density becomes critical, then some special things happen. So how this was relevant to the study of social insects, which I talked about in the beginning of my lecture. Well, now we finally get to bees. And let me start by introducing you what sort of data I worked with. And it is a very interesting data set. It was acquired by this guys from the University of Illinois. The lead author was Tim Gannard. You can check out the paper which describes the data. But they placed colonies of honeybees in the transparent hive, like a hive where one wall is made from glass. And then each bee, they attached a barcode to it, therefore permitting artificial algorithm to recognize each bee individually and track its movements and X and Y dimensions. And there was a PhD student who had to glue barcodes to thousands of bees. And I feel really sorry about him. But thanks to him, I had a very nice data to work with. And let's look at what I did. So now at this picture, you actually can see the movement of two bees in a hive. You can see that the trajectories are quite convoluted, and only two bees are shown here in blue and red lines. But obviously, there is like 1,000 bees in the hive, so they are moving in a. So it's kind of difficult to make sense of what is happening just from trajectories of single insects. So in order to understand it, we use a technique called coarse graining. So we have a hive, which is like 200, it's like 20 by 30 centimeters, so it's a rectangle. And we have the data for this hive. So then we kind of cover it with a grid, and we count the density of bees in each side of the grid. So obviously you can imagine that the sides on the corner, they would have little bees, but the sides in the center, they would have higher density of bees. And you can understand the logic. So here I just drew you a small example. So this side would have a density of 1 bee, and this side would have a density of 3 bees. And this is exactly what we can see on this heat map. We can see the edge effect, because bees are not visiting the edges. And we can see a high concentration of bees here, high density, because that is probably where the queen of the hive is. Because as you know, each hive has a queen, and this queen is capable of releasing pheromones which control the behavior of other insects. So they always tend to cluster around this larger B. But okay, so we have from this diagram observed that there are some inhomogeneities in density. So probably we can study how the fluctuations in density in different parts of the hive are correlated. But before we proceed to this, there might be some other quantities which we might want to measure. So as I told earlier, in the model of jamming, there is a control parameter, which is density, which controls what will happen on the road. So because bees are obviously located in a confined hive, and they are obviously interacting spatially, they are bumping into each other, they are sharing food, they are doing a lot of things, it makes sense that the overall behavior of the system would depend on the bee density. So, and the bee density is fluctuating, and it's fluctuating because bees can leave the hive, they can go out and forage, and because they can also go inside the grids of the honeycomb to sleep, eat, and do their usual routines. And in this picture, we can see the fluctuations in density. You can see that the recording took many days and nights, and during the day, the density is generally higher, and during the night, bees go to sleep. And there are also some other fluctuations which might be more difficult to understand on the stage. And at the same time, to have some sort of order parameter of how active bees are in the hive, we measure the mean speed of bees, like the mean activity of all bees in the system, which is here shown with black dots. And again, we can see that there are circadian rhythms in this quantity, that the bees are more active in the morning and less active in the night, and that also this speed increases as the time goes by. So these are for some of the order and control parameters which describe the system as a whole. But in order to have some more understanding of what individual bees are doing, we measure the inter-neighbor distance. So for each bee, we measure the average distance from the next bee in order to understand how compact they are. And we again can see some fluctuations here in both the mean distance and its variance. And then we can see that in the end, the mean distance increases. And that happens because in the end of the experiment, a lot of foragers had left the hive and they have never come back. So it became more sparse. So we can already see that some things are happening here spatially. But now, as I said before, the key feature of the critical system is the presence of long-range power law correlations. So we will try to infer these long-range correlations in the beehive. In order to do this, let's return for a bit to this very ugly diagram I drawn. So for each side, let's say we take this side and this side, we can have a time series with time going in this direction of how the density of bees at this site changes during the course of many days and nights. And then we can therefore measure correlations between these fluctuations of activity at different sides of the hive. And we can indeed observe that these are very long-range correlations, because these are spatial correlations between activity of bees at different parts of the hive. And you can see that on the x-axis, we have the distance, and the distance at which correlation acts, like the distance at which fluctuations of density at one side can affect another side, is like around more than 15 centimeters, while the size of an individual bee is 1 centimeter. So although two bees in the hive don't have a direct way to communicate, to push each other, their movements, the effect of their movements is affecting bees very far away from the bee which moved. And this should remind us of the fluctuations of magnetic dipoles, which we have discussed earlier in the case of the Ising model, and how the change of one magnetic moment can affect the magnetization very far away in the grid. In the same way, the fluctuations of density, of bee density at one side, can have an effect at the bee density at another side 10 or 15 centimeters away. So this type of long-range power law correlation implies that the beehive, in a similar fashion to the Ising model at the critical temperature and to the human brain, is existing in the critical state. Therefore, it's undergoing a phase transition. But what is the mechanism of this phase transition? And here we use the jamming model, the model of cars moving on the road, to explain how this happens. Just instead of cars, we have bees, and instead of road, which has a direction, we consider a two-dimensional version of this jamming model. So we imagine that we cover the entire beehive with the grid, and then through each grid side, we draw a line and count the number of crossings of this line in either direction to see how active is the flow of bees, how much they're changing their position. And then we plot this traffic, which is a quantity analogous to flow of cars I shown in the earlier diagram here, and occupancy, which is analogous to density. And we observe that initially traffic grows as a linear function of occupancy, implying that the more bees we have, the more bees are crossing these lines which are dividing the grid cells. But at a certain point, this trend reverses because too many bees are present in the sites. They are preventing each other from moving. They're bumping into each other, so they are getting jammed, just as the cars were. And in this jammed state, the small movements made by one bee start to affect movements made by another bee at a very long distance, because the whole system becomes interconnected, and you cannot move one part of it without affecting the whole system. And the colors here represent different phases of the experiment. And these phases correspond to different overall density of bees in the hive. Because in the beginning of the experiment, there were a lot of bees. And then in the end, many of them were allowed to leave and they just never come back. And this last state is corresponding to, marked by black dots. So we can see that the slope of correlations that descends much faster, meaning that there is less correlation in the system when the density is lower than while if we look at blue and brown dots, which correspond to the system with higher density, we can see that the correlations are descending much slower, therefore showing that indeed the density is the control parameter for the system. Now we compute what another quantity we discussed earlier, but then in the context of the human brain, the connected correlation function. So the correlation minus the mean value of activity in the system. And we can see that it is an exponential function and that it crosses zero, and the zero crossings, the correlation length, can be plotted against the size of the partition. So what does it mean? We can take an entire system and then we can compute a correlation function for a subset of the system. And then we can compute the correlation function for the larger subset of the system. And then again. And at each of the subsets, the correlation length, and the bigger is the subset, the bigger would be the correlation length, which if you think is rather counterintuitive, but it is what happens with the divergence of correlation length at the critical systems. So for the car traffic, we have seen that it is possible to create a model which captures some essential properties of the system. So let's see if it's possible to do the same with the beehive. So we created a two-dimensional model of the beehive, which is again a cellular automaton. And the bees are just represented as agents which can move in any direction within this system. But they are trying to avoid bumping into each other. And they're trying to avoid bumping in, and they're trying to move at their maximal speed. So we can have, we see here this model of beehive at 3 densities. And in the bottom layer, you can see just how this looks like if you take a snapshot, and the black grid corresponds to the grid which is occupied by an agent, and the white grid corresponds to the empty grid. And at the top layer, you can see the mean values of density. And you can see that at low and high densities, you don't have any specific structure, but at the intermediate critical density, you have this structure where you have a toroidal jam, like a ring jam of stack agents, which is marked here by a green color, and then inside it you have like a blue bubble in which a free movement is possible. So we compute the connected correlation function and the correlation length, which we discussed earlier, and we plot the correlation length versus density for different models. So we take a few dozen models with different densities of agents, and for each of them we determine the correlation length. And we can see that the correlation length grows with density until it reaches a certain critical value when it's maximal. And then it starts to go down, meaning that there is an optimal value of the density parameter at which the correlations in the system are maximal and the system is most interconnected. But if it's too low or too high, then the system becomes disjoint, the movement of agent becomes more independent. And this confirms our earlier suspicions about the jamming transition in the beehive and the idea that the density of bees is what is controlling how much independence they have in their movements and how much correlations exist in this beehive. And we can observe, again, this pattern that when we are changing the size of the model, when we are using the models of different sizes, then the correlation length for each of these partitions increases with the size of the partition, meaning that the small model is as correlated as a larger model. Now we compute the same quantity traffic, the flow of agents through the grid cell, and we can see that at the intermediate density we have this parabolic shape, which shows that initially as the density at the site grows, so does the number of agents which cross it. But then when we increase the density, the site becomes just filled with stack agents, and therefore no movement is possible. While at the high and low densities, we don't observe this parabolic shape. We just see a simple linear relationship. I will have some water. Okay, so I think that's it for bees. And now we are going to, as we still have some time, we are going to talk about a slightly different system, and we are going to talk about random walks and foraging. So are you familiar with the concept of animal foraging? Okay, excellent. Have you ever considered what is the best way for animals to forage? Okay. Well, neither did I until recently, but curiously enough, this problem has been quite big during the World War II, when the German submarines were going underneath the water to hunt for different ships heading with supplies from America to Britain or from America to Russia. And the Allied planes were trying to find these ships and, find these submarines and bomb them. So what they were trying to, they obviously didn't know where the submarines were, or they didn't back then have good radars which would permit finding something in the depth of water. So they would only spot the submarines when they would go up in order to take in the air. So they were trying to understand what is the best system to look for something if you don't know where to look for. May I remove this board for a bit and then put it back? Okay. Well, I'll have to draw on top of things, but I think it will be fine. But so let's say that you have a certain agent. It can be a plane, or it can be a wolf which is looking for its dinner. And then you have some randomly distributed pieces of prey, and you don't really know where they are. Like, you know that they have probably a certain density at this area, but that's all. So you need to kind of define a statistical rules for trajectory which your agent would follow. So which trajectory would you think would suit best? Would it be a good idea to go like this? Just make a lot of very short steps? Or would it be a good idea to make very long steps, like this, to cover a lot of area? Well, circles.
Spiral.
Yeah, so one thing, if you like have an actual plane and you have a lot of fuel and a lot of time, you might do what is called a grid search, right? Like just go like something like this. But if your subjects are moving and if you don't have much time, then you kind of need to combine the necessity to scout a lot of area and then to pay more attention to special areas where you might have something more of interest. Turns out that there is a special mathematical phenomenon called the Levy walk, which describes exactly this situation. When you are not exactly sure what you are looking for, and you are trying to combine the necessity to cover large area and the need to examine small pieces of it. And this trajectory would look like this. It would combine some very long steps, allowing you to switch from one region of space to another. And some very short steps when you are actually trying to locate whatever resource you are looking for. So if you measure the distance of these steps, the distance r, and if you plot the distribution of this distance, this is the probability of the step, and this is the length of the step, then you'll end up with a distribution like this, a power law distribution where you have a few very long steps and lots of very short steps. And turns out that it is indeed how many actual animals forage. So the first study was done on wandering albatrosses. So the albatrosses, they were not really foraging, but they were flying over water, and they needed to find themselves places to rest. And obviously not having an exact map of where these places are, they would adapt this Levy flight motion in order to locate them in the most efficient manner, simultaneously covering a lot of area and being able to find some place to sit down and rest. And this paper had an interesting history. First, it was disproven, then it was again proven as right. And there were a lot of mathematical tricks involved there. But what we can take away from there is that this Levy walk appears as an optimal foraging scenario where you have some sort of sparse resource distribution and you don't know its exact whereabouts, and you also have time constraints and stuff like this, which you would expect of a foraging animal. So it has been found in many species of animals, such as wolves, monkeys, and other animals which look for some resource, and also in human hunter-gatherers who are going around looking for wood and other materials. And curiously enough, it has not been so much studied in modern humans. And we had this idea that possibly if we consider a sportive game like football, then maybe in some aspect it would be very similar to foraging, because we have players who are trying to locate some sort of resource, and they're presumably motivated to do so, but at the same time they are operating under the temporal and spatial constraints. Can we actually have the screen back again? Okay, maybe I didn't need to draw this diagram that much, but interesting. Is it infinite? Like if we press it down, will it like infinitely go down? No? So here you can see an example of a random of a levy walk and a Brownian motion. Do you know how Brownian motion happens? Like when you have Brownian motion was discovered when Brown was observing pieces of pollen swimming in the water underneath the microscope, and he noticed that they are moving erratically. And later it was understood that this erratic movement is happening because small enough objects are colliding with water molecules, and these random collisions are propelling them to go erratically. And because they're so small, different number of collisions can come on one side and different number of collisions can come on another side, therefore unbalancing the motion of the particle. And if you have particle large enough, then a number of molecules which are colliding to the right of it and to the left of it would be roughly equal, therefore it would remain stationary. But this Brownian motion, which is characteristic of almost all inanimate objects, has a like if you measure the step length distribution between different bouts of motion, it will turn out to be exponential. And in case of a levy walk, it will be a power law distribution with this few long steps being towards the end of the tail, meaning that they're very rare, but they still happen. And as I've mentioned before, it has been found in this type of motion has been found in birds, human hunter-gatherers, and even in some extinct isopods, which are obviously dead, so they don't move, but their traces have been preserved in fossils. So it's possible to see how they've been moving billions of years ago. So we have studied if it's possible to perceive a football game as a case of foraging, the forage resource in this case being intuitively the ball, and the foragers are all the players in the team. And in order to do so, we had a very similar type of data as we have used in the last study I presented, the XY trajectories of different football players, which have been collected during the matches of the Japanese Football League during 1 year. Well, here you can see some examples of how they actually look like. Here is a trajectory of all players shown in fade blue and green colors, and the trajectories of centers of mass of the team, so like the mean position of all players for each respective team, being shown in bold colors. Here we measure the mean distance of all players from their center of mass. So if it's small, this means that the team is contracted, and if it's large, that means that the team is expanded and covering a large area. So we can see some interesting dynamics here which are happening on the collective level. But for now, we'll see what is happening on the individual level. So we'll take the trajectory of each football player, and then we'll partition it based on how often it's turning, and then we'll measure a distance of each successive step between different turns. And what we'll observe on this picture here, these are data shown for 4 different players, including the goalkeeper in the first diagram. And what we can see here is that most of these movements are minuscule. Probably they are movements when player is trying to get possession of the ball, or just moving to get a better sight of the target or something like this. But some of the steps are very large, implying that players are combining long and small steps in a similar fashion to foraging animals. Therefore, in a sense, you can interpret the game of football as a type of foraging, at least from the point of view of statistical patterns that arise here. But more interestingly, I think, is that you can apply a physical method to describe a random walk and then see how it can be relevant to a football game. And maybe the reason why football is so attractive to many people is because it somehow reminds us of our hunter-gathering past. Who knows? Finally, what I found quite curious is that earlier we computed the trajectories for the center of mass of each team, like an overall position of all players for each team. And if you compute in the same way the step size distribution for the centers of masses, you will observe that they are following a very similar pattern as individual players, this power law distribution, in which you have small and large movements combined together in different quantities. So I think it's interesting that the inherent coordination of the football team makes them replicate the individual patterns of movement on the group level. Right. I think that's all I have for you today in terms of what I said, but if you have questions, I would be glad to answer them.
Thank you very much. A lot of data. Hard to keep up with everything you said, but quite prolific, I think, for new ideas. And one question I have is regarding demographics. Do you think, I mean, talking about density, optimal density, do you think that this could be sort of applied to the overall population, world population, to find like optimal interaction?
Yeah, there is a lot of studies in terms of studying cities, trying to understand how people interact when they are gathered in like large conglomerates and cities, and several rules have been established. For example, that the bigger city, like if you have the bigger city, then the number of good things you are getting, for example, the number of inventions or the number of patents you are getting, grows faster than you would expect of the gathering of people of this size, meaning that one large city produces more inventions than 10 smaller cities with the same number of people, and stuff like this. Yeah, definitely, like a lot of this can be applied to larger demographic structures in terms of countries or cities.
Mm-hmm. Yeah. And of course, this takes us to the optimal point or the critical point that you sort of comes over and over again. And I think that it's a very interesting that transition phase and the turning points for many things, as you showed us. And regarding consciousness, because you did talk about that at the beginning, and brain. And you're talking about long range interactions,
correlations, yes.
right? Because in about 40 years ago, I remember there were studies about long range synchronicity. I mean, synchronicity in long range interaction between neuron populations. Do you think that could also be applied to that in order to sort of explain some kind of cognitive functions?
Yeah, it has been applied a lot. There's been a lot of studies studying in vivo neural networks, or in vitro neural networks, or in vivo brains, studying different synchronization and correlation between different parts. And it is implied that a healthy brain or a healthy tissue exists close to the critical point, while for like anesthetized state or for an epileptic state, you have departures from the critical point. Because for a brain, you obviously need to balance excitation and inhibition. Because if you have too much excitation, you end up having epileptic seizures, which take the whole cortex.
If you have too much what?
If you have too much excitation, if you have too much correlation, you can end up having epileptic seizures, which take all activity. And if you have inhibited state, like an anesthetized state, then you have too little correlation, and different parts of the brain are not interacting with each other.
Right.
So the critical point serves as a kind of balance between different needs and allows to have an optimal computational capacity. The tricky thing here is that the systems I talked about, the jamming of cars and the jamming of bees, there you can find a relatively simple control parameter, like the density. It makes sense. But when you are starting to look for a control parameter in the brain, things become difficult, because several candidates have been proposed, like some sort of balance of neurotransmitters or some sort of brain temperature measures, but they are obviously kind of difficult to come up with because the system is so complex.
Okay, thank you.
Oops.
Thank you very much. While you were speaking about the foraging,
Mhm.
I was thinking about this motricity cognition in bees, for example. And does this helps to how can I say it, to save energy and because of the movements the bees are doing for foraging in foraging, does this helps them to save this energy? And can I translate this in humans? I personally am working with thesis in Neanderthals. So in the past humans and the modern humans, does this helps to share how can I say it, a
Conserve energy.
spatial cognition, spatial consciousness of the places where you can save energy to move and to do this foraging.
Yeah, I think you are definitely on the right track here, because why the need for this foraging mechanisms arises, because while foraging, your aim is to get energy by yum yum, and you are spending energy by running, so you need to kind of conserve as much energy as you can while getting as much energy as you can. Otherwise, you could have just done a grid search or just go in a big spiral, as somebody I think suggested. So I think extinct human populations definitely probably have used some of these techniques to optimize their gathering of resources. I don't know about any studies of Neanderthals. There has been a study of modern hunter-gatherers who live in Guatemala, and they are foraging for firewood, I think, or some other commodity. And they are actually using this Levy walk paradigm in order to optimize. Well, when I say optimize, I don't mean they consciously do it, right? Like it arises from the need of optimization, but they are having these Levy patterns in their movements.
Thank you.
Hi, thank you for your talk. I wanted to ask you something about the big picture of what you were presenting.
Mm-hmm.
So mainly is what is the metaphysical claim between finding the same function in all those systems? Is it that the same properties emerge or the same properties are caused? Or what is the overall main aim? Because I was a bit confused when you were talking about the brain and then the bees, if that implies that to some extent there's a certain form of property that goes over and above the bees. I'm just questioning a little bit what is the main claim about finding that same pattern in all those systems.
Well, I think there are definitely properties which go above both the bees and the brains, right? Like if you are familiar with the term convergent evolution, like when we have similar evolutionary properties arising and disjoint animal groups, then I would say that all conscious entities, and I would say that both the brain and the beehive are in a sense, well, not conscious, but I would say cognitive entity. And I would say that both the brain and the beehive are cognitive entities because they are taking in information, they are processing information, and they are having to make decisions. I would say that all cognitive entities would come to have some similar organizational principles, like being in the critical state, for example, which would unite them. Yeah,
We should, sorry, what?
which would be shared amongst them if the media is different. I think the analogy would be the human brains and the artificial neural networks. Like they are quite far from each other, but there are certain similarities between them which came to be independently and despite them being in a very different material form.
And does that mean that they are both intelligent in a certain sense?
I think it's intelligent, yes.
Hmm.
I think the next question often asked is, are they actually conscious? Which I think no. But intelligent, yeah, I think it's fair to say this.
And do you think they have agency to a similar
Who,
extent as
bees
well, in
or
terms of as
bee-
far as they are both intelligent, they have certain like capacities of agency, or how far are you willing to go
with whom? With bees or
with the
with?
beast brain analogy?
Uh, I think with bees, I'm willing to say that they have a certain degree of agency because bees are very special, right? Individual bee does not reproduce. Only the colony reproduces as a whole through its hive queen. So they are, in a sense, evolving as colonies. And I would say that the colony of bees has a certain colony agency, which is emergent and somewhat more than agencies of individual bees. Yeah.
Thank you.
Well, I do have one question. So this is very interesting. So I'm wondering if maybe these patterns could be thought of as a mark of cognition or something like that. So if we identify certain pattern that is present in all cognitive systems, then maybe that is a mark of cognition. Do you think that could be the case, that any of these, I don't know, maybe being in the verge of chaos or any other sort of pattern, do you think it could be identified as a mark of a cognitive phenomenon taking place?
I hope so, yes, but I'm also quite careful with this. Have you heard of something called the integrated information theory? Well, if not all of you know, then integration information theory was brought up as being this universal signature of cognition, and it's based on like a complex decomposition of. Okay, anyway, like I'm not going to go into the math because it's actually quite complex, and at some moment when the authors were presenting this integrated information theory on a conference, another group of researchers came to them and showed them some sort of artificial neural network for which they measured this integrated information index, and it was higher than human. And so they asked these authors, like, so do you think this neural network is actually more conscious than you are? And curiously enough, the researchers, who was Tononi, like a very famous neuroscientist, said that, like, yeah, I do think it's more conscious than I am, which was kind of a strange thing to say. But the reason I brought up this story is that I kind of love the idea of having a universal metric for consciousness, but I'm also kind of careful with it, since quite a few had already been invented and not all of them have went well. Or like being them, many of them end up being like either too generic or too focusing on edge cases or something like this.
Okay, thank you.
Thank you. Well, as a follow-up on the question of agency and cognition, I was recently talking with Fernando Rosas, that you probably know. He works on complex systems, and he did mention that a mark of agency, cognitive agency, would be decision making, which you seem to endorse, right? I mean, from what you said now. But we do also philosophy here in the center, so this is more of a philosophical question. In order to have agency in that sense, meaning decision making, you need to presuppose some degree of freedom, right? Otherwise, it's not truly a decision, right? I mean, it's either a deterministic system or a to some degree open or free system. So how would you, what's your take on these systems you have shown, like whether bees or brain or whatever, are they deterministic? I mean, you did talk about chaos theory at the beginning, which are non-deterministic, but you mentioned, what's the word you mentioned? Well, anyhow, so what's your take about freedom and determinism in order to qualify a cognitive agent? Because some people talk about reactive agents and seem to make a difference between a reactive agent and a cognitive, truly cognitive agent. So I seem to think, I mean, intuitively at least, that cognition presupposes some degree of freedom. So what will you say about all that?
I think in what I think there are two layers. So on a very practical layer, for me, it is sufficient that when I do research, for me, it's sufficient to say that an agent can have goals and can have its behavior can have a behavior motivated by goals, and then I can say that it has certain agency. But this is like on a very low level. If the agent has these properties, I can do research on it. On a kind of higher level, I believe in what is called a strong emergence, that if you have a lot of very simple interacting nonlinear components, then out of them you can have an emergence of phenomena which are not explainable by studying these individual agents. So that is how I explain to myself the concept of free will and the concept of like complex behavior arising from physical systems. And like I definitely think that the human brain has enough complexity and nonlinearity in it to merit such emergent, strongly emergent free will. For bees, maybe very little. Who knows? I know that there are a lot of conflicting views on this topic. I'm not sure if you read the recent book by Sapolsky, Determined. Anyway, like the reason I mentioned this is that for me, I think it was kind of always intuitive to assume the existence of free will, since it is given to us in our perception so evidently that it makes sense to build our theories around it in order to understand how can we facilitate its presence. But I know that there is like an opposing school of thought which says that there is basically no free will, and we kind of need to just accept this. But I was never been able to kind of take it too seriously.
Mm-hmm.
I'm not sure what your stance is.
Mm-hmm. I think, I presuppose to freedom, as a sort of necessary trait in order to make sense of decision making and consciousness in general.
Well, yeah, if we remove the notion of free will, we will need to remove too many of our assumptions about the world.
About the what?
If we remove our notion of free will, we will need to remove too many of our assumptions about ourselves and the behavior of others. So we would end up living in a world which is actually entirely different. So I think it's a too drastic step to take in this stage.
Yeah, I agree. Thank you.
Well, I do have one more question about the Guatemalan foragers.
Mhm.
So how do you think they came up with this pattern? If it's not conscious, like how is it somehow genetically programmed? Are we like genetically predisposed to follow that Levi walk, or how do they arrive to it?
I think in all organisms it would be different. In some simple organisms, it would be, yes, just genetically programmed and then enhanced by the evolution. In some cases, it would arise naturally from interactions with the environment. So I think for these Guatemala foragers, the hypothesis proposed by the original authors of the study was that the structure of the landscapes in which they live kind of makes them, through the interactions with this landscape, follow this Levi walk pattern.
Okay. So it's in the highlands, or
No, they live in the jungle.
in the jungle. Okay. Okay. Thank you. Well, any other questions? No. Okay. Well, let's thank our speaker. I would like to give you this as a recognition for your talk.
Thank you.
There you go.
Thank you.
You're welcome. Okay, well, thank you.